will start the event with the introduction of IndoUSrare, followed by presentation by Dr. Partha Mukherjee, who is the Senior Director or Head of CMC Analytical Development, Amicus Therapeutics. The session will be moderated by Dr. Harsha Rajasimha, who is the founder and executive chairman of IndoUSrare. We will be having a thought-provoking discussion on enzyme replacement therapy on rare diseases, followed by closing remarks by Dr. Harsha Rajasimha. Before we begin today's event, I would like to give a brief introduction to our organization. IndoUSrare was founded with a vision to address the significant gaps in global diversity, equity, and access to clinical trials for rare diseases. We are a non-profit 501(c) charity organization uh, based out of Virginia, USA, and we work to combat rare diseases by fostering cross-border research, collaborations, data sharing, and clinical trials globally. The key problems we are trying to address is severe lack of global diversity in clinical trials and underrepresentation of the Indian diaspora in rare diseases. IndoUSrare aims to tackle this problem through our mission of building collaborative bridges to the Eastern and Western Hemisphere to increase diversity at the global level. Under this mission, we currently run 6 patient-centric programs, such as Patient Concierge, Patients Alliance, Research Program, Educational and Awareness, Advocacy, and Corporate Alliance Program. Our tech-enabled Patient Concierge connects and supports patients in need of clinical trials and supports patients in need of clinical trials, experts, and partners, and provides resources for patient education. Currently, we have received 150 plus patient requests all over the country, all over the continent, with over for 90 plus diseases, wherein top 3 requests included access to treatment, access to expert opinion, and connection with clinical trials. The other program is the Patients Alliance program, where we currently have a coalition of 66 patient foundations across 4 continents, which aims to connect patient groups and foundation in the US and their counterparts. I want to take a moment to recognize and highlight the incredible Patient Alliance members we are fortunate to collaborate with. Our research programs include our research code and various grant-funded research and education projects. And the next is our Corporate Alliance program, which aims to collaborate with pharmaceutical companies involved in rare disease research to ensure equitable and affordable access to therapies, which runs on 5 pillars. Currently, we have 5 members in this Corporate Alliance program. Every year, we also conduct various education and awareness events consisting webinar series, Rare Disease Day, Art for Rare, and Road for Rare, and participates in various conferences and other engagements. We are also involved in rare disease advocacy in US and India, where we have participated in various advocacy and events throughout these years. We proudly uphold various seals and badges for maintaining transparency and are honored to be active members of both Nord and Global Genes, where we continuously foster meaningful collaboration with the rare disease community. Over the 5 years, we have partnered with 66 patient alliance members, 5 corporate alliance members, who collectively represent over 500 plus rare diseases and 20,000 patients across 50 plus countries. Through our patient concierge program, we have hosted more than 25 global events and built on fostered over 20 partnerships, contributed to various policy inputs on data sharing and diversity in the US and India. Now, we would like to highlight some of the new initiatives at IndoUSrare to develop a blueprint for US-India Strategic Cooperation for Rare Disease Research and Orphan Drug. We have formed 4 working groups. The first working group 1, which is on newborn screening, diagnosis, patient registries, referral networks. The other one is the working group 2, which is on strategy to increase representation of Indian diaspora in clinical trials Working Group 3, which is based in legislative, regulatory, and financial advocacy, and Working Group 4, which is on affordable innovation, R&D, and manufacturing. If you're interested, please sign up for our working groups. We'll be sharing the relevant links directly in the chat box. The next initiative is the IndoUSrare flagship event, which is an IndoUSrare Bridging Rare Summit. Since 2023, we have been conducting this in-person event. IndoUSrare Summit aims to herald a new era of cooperation for rare diseases, orphan drug development. The 2023 summit was held on October 28th and 29th at JMU, Virginia, USA, and 2024 was held in November 16 to 18 at Indian National Science Academy, New Delhi. We look forward now to welcoming you all to this year's summit that is on November 3rd to 4th at the Hilton Performing Arts Center, JMU, Manassas, Virginia. This event will include keynotes, panel discussions, pitch for rare contests, workshops, posters and exhibits, ABMS Kushi Bridging Rare Awards, Bridging Rare Gala, cultural program, and ample networking opportunities. We invite you all to the event to participate in various ways by submitting a speaker and poster abstract and also applying for pitch for rare. We also invite you for Bridging Rare Gala, where we are featuring a BMS Kushi Bridging Rare Award and live performances by various artists. We'll be sharing the link for registration in the chat box shortly, where you can, register yourself for any of the events. With this, we move to the main event. And before that, I would like to share some of a few housekeeping points. Please keep your video off and microphone muted. And if you have any questions, put them in the chat box. We'll read them out as time permits. The recording will be posted online on all the IndoUSrare social handles. Now, I would like to invite Dr. Harsha Rajasimha, Founder and Executive Chair for IndoUSrare and Founder and CEO of Jeeva Clinical Trials, who will be moderating this session. Dr. Harsha, over to you.
Thank you, Ramya. Next slide, please.
Yes.
Yeah, it's my utmost pleasure to welcome you all to this webinar on enzyme replacement therapy, which is one of the critical and important treatment modalities for rare diseases, particularly when enzyme is missing due to a gene mutation or other reasons. And we have an expert here with us, Dr. Partha Mukherjee, who will be sharing his insights from decades of experience in the industry. Dr. Partha Mukherjee obtained his PhD in pharmaceutical analysis from Virginia Commonwealth University. Apparently, my daughter is just studying biomedical engineering in the same university this week. So, and his 28 years of illustrious career has spanned some of the major pharmaceutical companies like Pfizer, AstraZeneca, GlaxoSmithKline, Novartis, and so on. And his analytical CMC leadership roles have included cardiovascular, central nervous system, gastroenterology, and various disease areas, including smoking cessation, respiratory, and others. He has led numerous new drug applications, biologic applications, ANDAs, and commercial product launches, and with over 15 peer-reviewed publications and frequently invited speaker at national and international scientific conferences. He's been a advisor and a volunteer to IndoUSrare and helping shape the future for rare diseases, which is very fortunate for the rare disease community and for IndoUSrare over the years. Although Dr. Partha Mukherjee woke up sick this morning, he's still wanted to keep up his commitment. So I really appreciate Partha taking the time to share his insights with us today. And this webinar will be recorded and made available to everyone to learn from his expertise later as well. With that, I welcome and hand it over to Dr. Partha Mukherjee for his today's presentation.
Okay. Thank you, Harsha. That was very nice introduction. And yeah, it's a little bit under the weather, but I think I can, go through the presentation and we'll take it from there. So with that, yeah, it's a great honor, first of all, to speak on behalf of IndoUSrare. This is a great organization. That's what motivated me to join this organization, and working with Harsha and the dynamic team. It's been a pleasure so far. So I'm going to share my slides and take it from there. Okay. All right, hopefully you can see my slide.
Yes, we can see it now.
Okay. Okay. All right. So yeah, we have, I mean, this presentation is supposed to be very comprehensive presentation. We may run out of time. I'm giving you just a heads up because there are a lot of material. However, what we will do is we have the presentation material and this will be available to the registration, participants. So you will have that knowledge, base available to you. And if any questions later on, we can definitely deal with those. But I will be, based on the time left, I will be skipping some slides, that's for sure. So just to begin, this is not going to be any medical advice or discussion forum for the particular diseases. It's just an awareness session for lysosomal storage disorder, I will henceforth call as LSD. And there are various options available for treatment. I will try to focus on the ERT part as well, which is enzyme replacement therapy. And these are all going to be public knowledge, so there is no confidential information being shared. So just to begin with a little bit of humor in a way, LSD, as we know, if you type in Google, it will show up a agent which is a psychedelic, hallucinogenic drug substance. It's a, has been very popular in the old days, like in the 70s and things like that. So when you type LSD as such in Google search, it will inevitably show up LSD as this one. But what we are focusing on is the lysosomal storage disorder. Just a disclaimer. Okay, so let's begin with the very basics where we are starting. So lysosome, right? Lysosome is this one. It's a schematic on your left side. It's a spherical or oval shaped, sac basically inside our human cell, except RBC. And what it does is it has a membrane, phospholipid membrane, and inside that's called the lumen. In the lumen, it has more than 60 digestive enzymes. And those are called the hydrolase. If you see on your left, those are the list of various enzymes that are available. So you have the membrane, you have the enzymes. There is a proton pump, which actually maintains the acidic environment inside the lysosome lumen. And that helps to activate these enzymes. So there are a lot of transporter proteins and channels in this membrane. They actually bring in the nutrients, like if you have protein, carbohydrates, and fats, and then they get processed by those enzymes, and then they are trafficked back, through these membrane. And that's where the problem starts, which I will, show a little bit in few slides. So there are three major components, which is one of which is the hydrolase, which I mentioned, and their activators. Then you have proteins which are in the membrane itself. Those are structural protein. They are like channels for carrying the nutrition material back and forth. And then there is the lysosomal associated protein. So any abnormality that may happen with any of these proteins, that can actually lead to LSD. Okay. Um, then the way it works is the proteins actually get synthesized in the ribosome of our cell, that gets folded in the endoplasmic reticulum, and then eventually they get inside the lysosome. Okay, so anything that relates to the synthesis of lysosome, that can also cause an issue with the LSD. So there are various avenues which I'm trying to show where the LSD can start. So if you look at this pathophysiology, how this LSD works, so there are about 70% of the LSD, they're caused by mutation in lysosomal hydrolase, those enzymes, okay? And then you have a defective mechanism in the catabolite, which means the degradation product of your enzyme activity. There is a defect in the catabolite export getting out of the lysosome and the membrane trafficking for the nutrients coming through. And then there is also the chance of the undigested insoluble lipids, which gets, accumulated inside the system itself. So any of these can cause the LSD. What starts to happen is when these materials are getting, broken down by the enzyme, or if so, two things. One is if the enzyme is deficient or absent, then all the materials cannot break down whatever is inside the lysosome, right? The other thing is, even if they're broken down, if your trafficking system is not working, then they cannot get out of the cell. So in any case, what happens is your lysosome starts to expand in size, and that can lead to a lot of the problem, leading eventually to the organ malfunction, like liver, spleen, kidney, muscle, whatever the organ may be. There is a genetic factor. This is a genetic mutation of the enzyme that is responsible for breaking the various, nutrients or other substance inside the cell. There is the chance that basically you have to have a defective gene in both the parents, and that's how it starts. And then I'm not going through the details, but eventually there is a 1 in 4 chance of developing in the lysosomal storage disorder. That's what happens. So effectively, this is purely a genetic mutation factor that actually triggers the formation of this, LSD mechanism. So a very interesting thing, this LSD, this is very focused on the ethnicity of the population. For example, you will see some listing here. Some populations are very susceptible to few enzymes, like the Jewish population in Gaucher disease, etc. The Chinese and Asian population and African, they are very susceptible to Pompe disease, which is another LSD. So there are a total of about 70 LSD in the system that can happen because of these genetic defects. Okay. And so that's basically an issue. And then the way it's classified, the classic way of, defining the LSD is based on the substrate, what gets accumulated inside the lysosome because of the defect in the enzyme. Okay. The protein is actually listed as the enzyme here. And then there is the associated, mutated gene. So this is, as of now, this is the most comprehensive way of categorizing LSDs based on the biochemical nature of the substrate. So for example, sialic acid. If the sialic acid is not getting broken down by sialin, then it leads to this problem of celiac disease, which is an LSD basically, for lack of words, sialuria. Okay. Um, and then there is also a lot of other things. Glycogen, if it gets accumulated in the system and you don't have this enzyme GAA, then it leads to Pompe disease. And that's something I'll be touching upon later on. So that's kind of the snapshot. Each of these will take probably, full day for talking about these diseases. And then the clinical category is basically there are 7 categories of LSD, and that is decided based on the type of disease. So sphingolipidosis, that's because of the complex sphingosine containing phospholipids. They accumulate in the system, right? Then oligosaccharidosis, etc. The very big names. MPS, that's another very common, LSD, and the list goes on. But again, this is a very nice reference what are the different category based on clinical, classification of LSDs. So the most common LSD is Gaucher disease, that prevalent in 1 in 40,000 people. And then the Fabry disease follows the next, that is between 40,000 to 170,000 people. That's again deficiency of a particular enzyme. And then the other ones are listed here also, MPS 1, MPS 2, Pompe disease, etc. So main challenge is we don't have a very clear picture of these diseases just because we don't do the newborn screening on a routine basis in all the countries. The genetic database is not that robust. A lot of the disease goes undiagnosed, which is the problem with rare disease. The disease condition itself can be varying. For example, in Pompe, there is something called infantile onset Pompe disease, IOPD, and there is something called LOPD, or late onset Pompe disease. So it varies widely also, and that makes it difficult to get the accurate estimate. The ethnic variation also, what I showed. So there are a lot of factors which really doesn't help much to get the accurate estimate of the prevalence of these diseases. So I just wanted to make that very clear. So the general symptoms is it's going to affect your different organs, as I mentioned, heart, spleen, liver, muscle. There will be muscle weakness. Eventually, there can be heart failure. That's the ultimate, culmination of the LSD. Damage to the central nervous system, kidney failure, and the list goes on. And here you can see all the damages it can do, based on the pictures, the snapshots captured. It's a very dangerous disease to begin with, this LSD. So diagnostic test, fortunately, there are 3 ways we can diagnose it. One is during pregnancy. There are some tests available for that. The doctor can do that. Then for newborns, there is also possibility to do the blood test, and that is basically on the LSD right there. And then there are other testing like the genetic testing, biopsy, urine test, etc. So there are various ways. The system is getting more popular how we can diagnose the LSD, category of rare disease much earlier, even during pregnancy. And then there are tests, for adults and infants and so forth. So there are various categories of LSD treatment available. The enzyme replacement therapy, which is basically ERT. Simply you're administering the enzyme every 2 weeks by IV, and that treats a lot of different diseases like Gaucher, Fabry, MPS, Pompe, etc. So yeah, it actually helps in the cardiac and lung function, and it can make the quality of life much better, but it does have a lot of disadvantages. So I mean, I would say not a lot, but several disadvantages, which I will touch upon. The other ones are something called pharmacological chaperone therapy, which is PCT. What PCT is doing is basically, if your enzyme, it's a tertiary structure, if it gets folded, then it cannot work properly. So this PCT is actually going, attaching to your enzyme molecule, and it's making sure the enzyme is not getting misfolded, and that's how it maintains the integrity of the enzyme. And the example is migalastat, Galafold, for Fabry disease. The other one is proteostatis regulator, PR. What they do is they're not going to actually bind to the enzyme, but they actually shift the degradation pathway away from the enzyme. So that's another way of controlling or maintaining the functionality of the enzyme. Okay, so there's the aramicol, that is for Niemann-Pick disease. And it's actually, there are few which are in the clinical trial right now. It's gaining popularity, but it's not that popular yet. Then substrate reduction therapy, which is SR2, what it does is it's going to reduce the substrate on which the enzyme was supposed to work. So it's actually not touching the enzyme. It's actually reducing the load of your substrate inside the cell. So that's another way of tackling with the LSD. So the drug miglustat is very common for that in the Gaucher disease. Then the fifth category is the small molecule assisted substrate transportation SMAST. So what this is doing is, okay, the substrate that is inside your cell, it has accumulated, let's say glycogen, is not being able to come out from the cell because your transporter is lacking. So what this category does is they convert that to a system or a compound which can go through your transporter system. So that's a advantage is it's very helpful. You can give it orally. It gets biodistributed. It can cross the blood-brain barrier, BBB, so it can create neurological symptom treatment, which ERT cannot do, by the way. Okay. Then you have the anti-inflammatory agents, which are basically various kind of NSAIDs, as we know. Then there are all this listing of different compounds which are already in the clinical trial right now. It's being evaluated. It's not in the market or anything, but it's being very heavily focused because they do show improvement in terms of the brain inflammation, and the survival rate of the patients can be increased. Okay. Then another one which actually was in place before ERT showed up is the hematopoietic stem cell transplantation. That's called HSCT. So what we do is take a donor's stem cell, and that gets infused inside the patient who doesn't have that enzyme. So that donor cell actually start producing your deficient enzyme. So for example, in Hurler syndrome and various other disease, that's how the LSD treatment is working through this HSCT process. It does have some disadvantage, which I will touch upon, but it was there even before ERT showed up. And then gene therapy. Gene therapy is again getting a lot of attention. Essentially, this is an ex vivo autologous method. Basically, you take out patient's blood, you separate the target cells, and then you introduce a corrected version of the gene inside that cell, and then reintroduce that, infuse it back inside the patient. So this can be actually very long-term treatment, lifelong in some cases. That's one of the advantage of the gene therapy. And again, there are various treatments happening which are which are in the market or actually undergoing clinical trial in most cases. And then manipulation of gene therapy. This is mainly focused on the genome editing. I'm not going through the details in the as interest of time. Then the overall landscape, you will think ex vivo is where you're taking out the cells and introducing the corrected gene, ex vivo. In vivo is where you're directly injecting a viral vector, and that has the encoded gene of interest. So this is the ex vivo and in vivo. This is a little bit busy slide, but I wanted to show that there is tremendous amount of activity happening in the gene therapy landscape for treating LSD, as you can see from the list of ex vivo and in vivo, treatments which are out there. Okay. Ancillary care, it's actually going beyond the actual, drugs, but it gives a lot of other supplemental support for LSD, like medication for seizure, etc. Medication for endocrine dysfunction, not directly related with the LSD, but the treating the associated symptoms. So there's a huge list of those things. And this is just a snapshot to show the complexity of LSD current and future therapies. If you get some time, later on, just take a look at each of these, pie chart, segments, and it actually explains it very well. I don't have the time today to go through each individual, but it does capture, the process and mechanism very nicely in just this pie chart. So small molecules, they have a lot of potential in the LSD. Why? Because Number one, it's not going through IV route. The second thing is they can reach, because of the small size, they can cross the blood-brain barrier, reach your CNS system. So a lot of neurological LSD can be treated by small molecules. Less immune reaction also from the patients. And the various, modules of LSD, PCT, SRD, SMAST, and PR, which I just mentioned. The other good thing is one of those drugs can easily treat multiple LSD. So that's a very plus point of these small molecules in terms of LSD therapy. So one, particular small molecule drug can be used for treating, as you can see here, at least 5, 6 different drugs, disease conditions. So that's considered a huge benefit of small molecules compared to large molecules. So very quickly going through the various, advantages of LSD. So ERT, it's actually intravenously, you're giving a recombinant version of the deficient enzyme, which will allow to metabolize the accumulated substrate in the lysosome. That's it. So it can be targeted to specific tissue. It can be in multiple disorder. You have got clinical success, rapid action of that, and effective mainly for non-neurological symptom. SRT, which is the substrate reduction therapy, that is primarily again taken orally, which is very convenient. It can cross your blood-brain barrier. It's non-immunogenic, so that's another thing, and it allows continuous dosing. So the level of fluctuation that happens with ERT typically, that will be absent if you are using SRT. Then the stem cell transplantation, again, it can penetrate your CNS system. It has a very lifelong, it can be a lifelong source of the missing enzyme, broad efficacy, etc. Then in terms of gene therapy, you have the viral vectors designing, that delivering the functional copy of the gene, as I mentioned. BBB, overcoming, that's again another advantage of that. And it can be customized. That's the other good thing. So it can lead to patient-specific, therapy treatment for LSD because you can manipulate the gene in various ways ex vivo. So that's really a great advantage of that. For the pharmacological chaperone, remember the molecule which actually prevents the misfolding of the enzyme. So oral administration, it gets widely distributed in various organs, so it can actually address a lot of the organ-specific conditions. It's non-immunogenic. Then combinatorial therapy is basically, for example, I will give later on Pompe disease. They have Pombility, which is a large molecule combining with Opfolda, which is a small molecule. So that kind of combination therapy is gaining a lot of interest these days. Okay. Drawbacks. So ERT I will discuss later. SRT, their main drawbacks is they can cause various side effects and drug-drug interaction. So that's the problem. Then the PCT, which is pharmacological chaperone therapy, that works for only certain patients which have specific mutation. The efficacy can vary. Yeah, the clinical efficacy is yet to be proven for some of the, LSDs. Whereas for HCT, the main drawback is high risk procedure. It's organ failure can happen because of the stem cell, not fitting with the patient. Limited effectiveness for existing damage. Then donor limitation, that can be another thing, finding the right compatible donor. That can be a major roadblock for HSCT. Okay. In gene therapy, immune response is the main challenge. So that can potentially limit the treatment options. There could be unknown long-term side effects. That's very common actually in the gene therapy if you're giving ex vivo gene therapy, for example. And sometimes it can cause some irreversible change to the cells, which can be detrimental. So again, the bottom line is all of these therapies, they have their pros and cons, right? So it will be the doctor who will make the right decision to choose which available market, drug from these categories will be the best fit for the patient, depending on severity, running various, diagnostic tests, they can make that decision. So coming to ERT, ERT actually, came into the picture in terms of approval back in 1994. First time FDA got that approved, the first treatment. It's kind of the gold standard for treatment of LSD. It's been around for more than 2 decades. And There is a kind of misconception between enzyme replacement therapy and enzyme enhancement therapy. So, bottom line is enzyme replacement or ERT is where you don't have the enzyme or it's deficient, right? So, that's where it works. And the enzyme enhancement is yes, the enzyme is there, but it may have only some residual action left in that enzyme because of the misfolding or some unstable nature of the enzyme. So EET actually works where the enzyme is not completely non-functional, and it helps in, BBB penetration. It's actually leveraging patient's own protein, so you don't have to give an external enzyme, to the system. So that's another advantage. So as of now, there are about 15, and I'm not going through all these details, but about 15 to 20, ERT drugs out there. And the market last year, that was around $10.5 billion. So you can imagine that. And here is the list of FDA approval years. The first one was in 1991. The latest one was in September 2023, which happens to be our product from Amicus, actually, Pombiliti and Opfolda, that combination drug. So drawbacks, the ERT has several drawbacks. One is the short half-life. The half-life can be as small as 40 minutes only. So what that means is you have to give the infusion more frequently, right? It can cause a lot of immune reaction. The cost can be also, the other thing is it doesn't get into the blood-brain barrier or, poorly vascular tissues like bones. The cost can be very expensive, up to 200,000 US dollar annually for a patient. The efficacy can vary a little bit. That's another challenge. And it can be time consuming, meaning once you start on this, that means you are actually stuck with it. You have to have it lifelong therapy for that. Okay. Let's keep that. So for Pompe, basically, this is what I wanted to showcase as a case example. What it does here, you have in the chromosome 17, there is a gene which is responsible for acid alpha glucosidase, gene that's called GAA. That actually breaks down your glycogen to glucose. So if there is any mutation in that, that function is not happening within the lysosome. So what that means is you are accumulating glycogen inside your various, organs, specifically muscles, that gets the most affected. And that will lead to your muscle decay. If that also happens in the liver muscles and, heart muscles, then your heart and liver, everything, eventually there is organ failure. That's the main thing. So it's very serious disease, happens between 40,000 to 60,000 individuals, one in 40,000 to 60,000, because it's rare disease. But this is one of the common disease, probably in the ranking, it falls like third or fourth most prevalent rare disease right now. So the treatments are basically, it started with a compound called myozyme, a drug actually that was approved in 2006. It was from Sanofi. Basically, it's a recombinant GAA that gets injected in the bloodstream and that replaces the deficient enzyme. That's the way it works. But there are a lot of disadvantages of that because once it gets in the bloodstream, it can get very easily cleared. There was no mechanism to retain it in the bloodstream before it actually gets in your target, organs like the muscle tissues. So what Sanofi did is they came up with the second generation of ERT, which is Nexviazyme, and that got approved in 2021. Okay, and that was for something called a late on stage Pompe disease, for basically for adult population. What happens here is this drug now has a engineering done where there is something called bis M6P or mannose 6 phosphate that gets binded to the drug molecule, and then it allows that whole molecule to get very well bound to this receptor, which is called CIM6P. It's a cation independent mannose 6 phosphate, big name, receptor. Once it gets bound to the receptor, then it gets inside the lysosome and it starts to work. So this is a second generation of this drug basically. And this is how I mentioned, you infuse the drug, it gets bound to the receptor on your muscle cell, it gets inside the muscle by endocytosis, then it gets inside the lysosome where it breaks down the glycogen, it hydrolyzes the glycogen, converts it into glucose, and that's it. Okay. So the latest one, as I mentioned, this is the Pombiliti and Opfolda combination, glucosidase alpha. So the main advantage of this compound is it has two of these bis M6P molecules. So it actually helps to bind much stronger compared to the previous generation drugs for ERT for Pompe disease. So the other, advantage of this, it has a small molecule Opfolda, and that actually allows the drug to be stabilized when it's going through the bloodstream because it binds to binds to Pombiliti, and then it protects it from, getting degraded inside the bloodstream itself. So both of those, the BSM6P and the Opfolda, that combination makes it a third generation, basically ERT, and that actually gives you much more benefits compared to the competitor drugs. Treatment regimen, you start Opfolda, but then you also need to start the Pombiliti, which is the infusion stuff. So Opfolda is the oral capsule, then Pombiliti is your actual infusion. So that's kind of the schematic of the dosing regimen. As I mentioned, I think we can skip the mechanism. You can see Opfolda is getting bound to Pombiliti inside the bloodstream. This has those antenna kind of receptors that actually binds to your the receptor on the cell in the muscle cell, gets inside the lysosome, then Opfolda separates out, and then these accessories, they also separate out from your Pombiliti molecule. Then it binds to glycogen and starts working on it and converts it to glucose. That's kind of the summary of what it does in the body in few hours. So in terms of future, is basically there's a next generation of ERT where there is something called pegylation. So it actually stabilizes the ERT molecule, so you can have a much longer half-life, like almost 80 hours compared to 40 hours. There is antibody tagging that can happen. You can modify the enzymes so that they can cross the blood-brain barrier. There is something, a drug out there, which is still going through, I think, clinical trial, part GUS. So although there are drawbacks of ERT, there are a lot of research happening on the academia as well as in the industry to make it more rugged and more accessible to the blood-brain barrier, for example. So direct tissue administration of ERT, that's another future of ERT where it can get directly infused to your vertical, the ventricular, cerebroventricular space and intrathecal space. So it's basically bypassing the blood-brain barrier, and that way the enzyme gets directly absorbed inside the cell. Then the bone targeting also, so that falls under direct tissue administration. Nano encapsulation of the enzyme, that's another thing. If you go through this, in your convenience time, you will see there are a lot of research happening right now for nano encapsulation. So liposomes, nanoparticles, micelles, virus-like particles, a lot of different kind of carriers where the enzyme is getting encapsulated or somehow modified and it's getting delivered. Okay, so those are the advantages, but it's undergoing a lot of clinical trial right now. So coming to the conclusion, I think, in terms of the whole process, the LSDs are inborn errors in the metabolism, and they're caused by some genetic mutation. Primarily the lysosomal enzyme, if they're defective, that's the main thing. As I mentioned, like 70% of the LSDs are caused by ERT problem. And what happens, the commonality is the substrate is getting accumulated inside your cell. They cannot get out, and then it's causing some toxicity effect. There is a cascading effect. Now the cell is expanding, the organ is, expanding, the organ failure happening, and that's how the, LSD, gets its ultimately even fatal symptom will show up. The earlier the onset, the more severe the dysfunction, like in infants, it happens. Very, very difficult in terms of the mortality rate. The earlier the diagnosis, obviously, the better chances you have to deal with it. So newborn screening programs are now happening, and that could be the answer to detect LSDs early and gain the maximum therapeutic advantage. Yeah, I mean, that makes sense. The earlier you can diagnose, you can think of what would be the best course of treatment out of the 9 different therapies that I mentioned. So this is mainly the thing. And then just to wrap up, if you want to learn a little bit more about how CMC fits into the, ERT, that's a whole different, aspect, and we can actually talk about that in a future webinar. I mean, that could be a good candidate talking how the CMC fits in the ERT. But today's session was mainly focused on to give you a glimpse of the complexity of the LSD, how it happens, what are the different kinds of categories, of this LSD, and also what are the treatment options available out there, what are the pros and cons of those. So I tried to give you a very overall good, background on the ERT, LSD, and how it works, etc. So the slides again will be shared. So take your time to go through them and, be more aware on those things. So with that, I would like to conclude my talk. We have 10 minutes left, and I turn it over to Harsha for any comments or anything, and folks who have joined today. But it was a great pleasure and opportunity to present. I was more excited because I work for a rare disease company and, showcase the latest ERT that's out there, which is that Pombiliti Opfolda, and again, highlighting all the advantage and disadvantage of them at this time. So with that, I will take a break and open up for any comments, questions.
Thank you so much, Partha. It's very insightful. Learned a lot and a lot of new things that you presented today. Then I have seen your presentation earlier also, but very insightful. And this book chapter, I encourage everyone interested in learning more on the CMC manufacturing side, but also unique aspects of conducting clinical trials and research or drug development for rare diseases. It was great to collaborate with Partha on this book chapter that we co-authored. That's available now through Springer. And if anyone has any questions or comments, feel free to unmute yourself. And now it's time for question and answers. We have about 10 minutes. But I can get started. Partha, what do you think is the future of enzyme replacement therapies moving forward?
Yeah, going forward, that's a good question, Harsha, because there's a lot of research happening. A lot of these rare disease focus is coming back to the industry. Even some of the big pharma, they are now catching up on this rare disease space. So what they're looking for, they are doing a pros and cons analysis of the current portfolio of drugs that's out there. And the focus is going to be number one on small molecule based ERT, and that can be pharmacological chaperone therapy. It can be, the substrate reduction therapy, or also the small molecule assisted, therapy, that kind of thing. So one of the key focus in future is going to be on small molecules. As you know, enzymes being a, biological substance, so it's these are the large molecule space. So pumpability, for example, is a large molecule, but small molecule is one area. The other area which I touched upon is nano encapsulation. So that way the drug can be protected while it's going through the bloodstream, and how to have those nanoparticles actually go through the blood-brain barrier for more CNS related LSD treatment. So that's another area. And then the third thing is, as you know, I just touched upon, a lot of these current ERT, they are based on IV infusion, and they have a short half-life. So what the plan is to have some kind of conjugation of that, replacement therapy with small molecules, for example, so that it can increase the half-life of the drug. Um, for example, this pumpability, coming back to that because I know that much more, that has a half-life of 2.5 hours compared to 40 minutes for some of the other compounds. So that's another way we are trying to get that. And the real benefit will come from having a robust system for newborn screening, a more genomic based database that will help to capture the different scenario based on ethnic prevalence of ERTs, and how to address those. I think those will come very handy to have a better prediction of which disease is how much prevalent in globally, for example. We don't have that picture. So that's also another area where the future of ERT is coming into play, and a lot of the organizations, they are working on it to pull together those information in nice database. So I think the future is very positive for ERT, although right now it's only a short segment of the whole drug development scenario. But the good thing is bigger companies are also focusing on it, on the ERT space. So I think we have a good future of this, going forward.
Yeah, absolutely. Thank you, Partha. Anyone has any questions? Feel free to either raise your hand or unmute yourself, and feel free to speak or ask a question.
And again, I will share the slides, and there are some good supplemental information even in the backup slides. You feel free to go through those. I think that will be also helpful if you
Yeah,
want to learn more about these things.
absolutely. We'll be sharing this deck and the video recording with everyone after this webinar. Yes, Bijia, you have a question. Please go ahead.
Yes, thank you, sir, for such a wonderful presentation. A lot of insight about the topic. I just
Thank
had a
you.
question. In low and middle income countries, access to this enzyme replacement therapy remains a major challenge. So, how should a clinician and researchers in low resource setting go about recommending enzyme replacement therapy to patients? I mean, where can they find reliable sources for ERT and the practical steps that can be taken to ensure that patient actually receives it?
Yeah, that's a very good question from the logistic perspective, and that's what Harsha and the organization is actually, actively working on, how we can expand the scope of these rare disease drugs also to the LMICs, right? So I can tell from, experience with some of the rare disease companies that they are getting more and more interested to also expand the clinical trial also, in the low middle income countries, and provide and work with the government if possible to provide a subsidized rate of these, treatment. Because if you think about it, the rare disease companies, they are there for a reason. It's not that much profitable area compared to the big pharma, what they work on the various therapeutic areas. But they have this noble mission to expand to the population which are underprivileged. So there are discussions happening in the rare disease space within these organizations, how they can partner with other, non-profit volunteer organizations on those LMIC. There is also initiative to work with World Health Organization, for example, to see if there can be any leverage in that to, get these medicines access to more population, especially on the LMIC countries. So it's not gaining that much momentum, I have to say, yet, but the focus is happening right now. And hopefully, with the changes in the law, where there will be not that much taxes and things like that happening on this import of these drugs, which will be, if they can be considered a true orphan disease drug and have those advantages, I think that will play a big role for these drugs. um, in the clinical side also, we don't get the full picture sometimes, just because the clinical trial doesn't get extended to those countries, right? However, this undiagnosed or ethnic population is focusing on those areas. So there is a huge untapped population for each of these rare disease drugs, which we can definitely work on. And that's where I think our organization here, as well as the partnership with the interested companies, that will help out. So bottom line is, yes, it's the nice ambition is there to help out and get in the LMIC, but it hasn't got that much traction yet. I don't know, Harsha, if you have anything on that, because you'll know a little bit more.
No, thank you, Partha. Absolutely. We are looking at that. There is a lot of challenges in the launch sequence of any drugs. They first get launched in US, then Europe, then Japan, and then there is a long tail before they get to the rest of the world.
Exactly.
And that's one of the problems we are trying to address, collaborate, and it takes a lot of collaborations with multiple stakeholders. We absolutely need government policy makers at the table, but also industry leaders. If there is a will at the senior top leadership at these companies, there might be ways to make way for this. And the biotechnology industry organization Bio can play a big role in that, and as well as global collaboration. So Bio also has Bio Asia now, which is hosting conferences in the Asia Pac region. And at IndoUSrare, we are collaborating with all these institutions, and the working groups, number 4 especially, is looking at that particular aspect. So I encourage you to get involved. I know, Bijaya, you're already involved in getting involved. So that will be a good place to bring this up and get all the right people to join those working groups, contribute, and see how we can actually operationalize this. there have been limited success stories in that direction. For example, with compassionate use programs, expanded access programs, named patient basis access, stuff like that. So there are ways in which even in the past, the patients in India and other LMICs have accessed ERTs without necessarily patients having to pay out of pocket or pay reasonable amounts, affordable prices. But yeah, a lot of work needs to be done in that. So very important question.
Yeah, we have a long way to go.
And I just had one point to make. I mean, it's not only getting these enzymes available, but also there is an infrastructure that need to be built, because it's not easy to administer these therapies. You need to have hospitals that are trained, the staff is trained, the facility is up to date for administering these medications. And many times you need home infusions because patients are not able to travel. So that is something I feel like the manufacturers can work with these LMICs to train the workforce. I know Centogene was doing that in Pakistan to help with diagnosis, but no, I have not seen a company come forward doing that for administration. So maybe Amicus or any other of the rare disease companies maybe need to put in resources to train the workforce in LMICs because therapy, you can bring in the therapies, but if you are not able to get it to the patients, again, I mean, you can have charitable programs and all that, but if you don't have the workforce, it's not going to be helpful. So that is something we need to start talking early on.
Absolutely. Yeah, that's a good point, because in all the cases, the patient, they will have to travel, even like here, they have to travel to the clinic, and then they get admitted for a few hours, they get infused, and every 2 weeks they have to show up in the clinic, right?
Yep.
And then they have to follow very rigorous fasting regime or whatever the case may be. So a lot of this education and the infrastructure is very important to actually administer the IV based ERTs, which most of the drugs are right now, unfortunately.
And I just feel like the role of the pharmacist needs to be shown, or I mean, that should be kind of showcased and highlighted in these administration, because right now it's the physician trying to do everything with the help of the nurse or the hospital. But here, I mean, there are pharmacy services that does these home infusions. So pharmacists have their own workforce that they can go to the patient's home and do these infusions every 2 weeks. Take care of any side effects. They are trained to take care of adverse events, all that. So that is not done in places like India.
No, it's not there. Very limited. So there are, definitely, we have a long way to go in terms of setting up the infrastructure, getting the buy-in from government and other, relevant, agency.
Yeah. It's a chicken or egg problem, right? So what comes first? Is it education, training, and infrastructure, or is it the will to actually make the drugs available in those settings? But yes, that's a conundrum we have to solve.
Yeah, and that's how it works, unfortunately, or whichever way we call it. The drug actually gets launched and approved in primarily all this other countries, US, UK, LATAM, and then Asia, very few countries. Japan, for example, they will get that privilege. Then comes the LMIC, like the African countries and things. So we have some discussion, continue on this topic.
Yeah, thank you so much. Go ahead, Reena. I will let you have the last word.
No, I just wanted to congratulate Partha for Dr. Mukherjee for concising this topic. It's a very intense and vast topic, and you were able to bring it to a 1 hour webinar or a 45 minutes webinar. And I see the comments, people have found it very useful. And hopefully this is the start of the discussion. And as you see, if we could bring in the CMCs for ERTs as our next topic, and then talking about how do we educate or how do we train our workforces in this LMICs could be a subsequent session. So this is just the start of the conversation, and we have a long way to go.
Completely agree, Reena. Thank you. Absolutely. I think both complex topic and a lot of material was very nicely covered in a short period of time, which even I was able to understand, even though I'm not from that background. So thank you so much. Thank you for sharing that. We look forward to collaborating further.
Okay.
And I
Thank you
invite
all.
everyone to join us at the summit in November, where Dr. Partha Mukherjee and many others will be there. So you can interact in person and
Yes.
also join virtually in case in person is not convenient.
Thank you all. This was a very great experience. Thank you.
Thank you. Bye.
Bye.